So in this video, the type of pathogen we're going to talk very broadly and very quickly about are viruses. I just had to put this quote up that my genetics professor said in the late 90s. Viruses aren't bad, they just want to make babies. Absolutely true. The sole purpose of a virus infecting a human, an animal, a bacterial cell, whatever, is that it simply wants to reproduce itself. A virus is a biological entity. It is not a living thing. It cannot replicate itself independently. A virus is an obligate intracellular parasite. It absolutely relies on a host cell in order to replicate and be viable. The nature of the word obligate means that it has no other option but to live that lifestyle, so to speak, because viruses are not living things, once again. They're made of biological macromolecules. They have a nucleic acid core, they have a protein capsid, they're made by biological processes, but they in and of themselves do not direct biological processes. They don't have the equipment to do so. If you wish to have a reminder of this, just give a quick look again at the flu virus video that was also posted in conjunction with today's lecture. So just to give a quick overview of the life cycle of a virus, if you have a host cell here and you have a virus here, this virus is first going to attach to this host cell. It's then going to inject its nucleic acid. By and large, there are also some instances where this attached virus might be engulfed by phagocytosis into the host cell. The key point here is that this viral nucleic acid somehow has got to get into the host cell. Once it gets into the host cell, the nucleic acid is replicated. The nucleic acid is also read and expressed by the host cell. That nucleic acid is going to encode proteins that the viral particles are going to need in order to replicate themselves. So that would be things like the proteins that make up this little I am completely losing my train of thought. This little housing right here that protects the viral nucleic acid. This is called a capsid. We'll get back to that in a second. Once all of these capsid proteins and other proteins are expressed, once the viral nucleic acid is replicated, these are then reassembled into new viral particles. This is called packaging, and then the viral particles are released. If you get so many that they actually are released out by bursting the cell, that's called lysis. In other cases, these viruses actually exit the cell by simply budding out through the membrane. Okay, so a virus is pretty simple and consistent with what we said about bacterial cells. There are only a few things that every single virus has, regardless of type. These are not the only components of a virus, but these are the two that all viruses have. All viruses have a genome. That would be a piece of nucleic acid, sometimes multiple pieces, that can be either DNA or RNA. It's usually just one piece. It's usually linear as opposed to circular DNA, which is what bacterial cells have. When a viral genome is only one piece, we call it non-segmented. When it's multiple pieces, we call it segmented. Viral nucleic acids are covered and protected by something called a capsid. This is the thing I had a hard time coming up with an alternative word for a second ago. I just wanted to call it a capsid. A capsid is made up of these repeating individual units of the exact same protein. They're assembled with the viral nucleic acid in the middle. Many viruses, not all, have an envelope around the capsid. So, in some cases, this is the entire virus. In other cases, you have this protected by an envelope. So, this envelope is actually going to be made of, and in fact derived from, the exact same material as the membrane of the host cell that made this virus. What I mentioned a second ago about progeny virus budding out through the cell membrane, when they do that, sometimes they take a bit of the membrane with them, they coat themselves with it, and that becomes a viral envelope. So there are some more virus parts that are, in fact, even more specialized than something like an envelope, and there are a few that are definitely worth mentioning, in particular because they are excellent antiviral drug targets. Some of these include, broadly, things like enzymes. And specific enzymes that I want to mention are something called lysozyme. So this is an enzyme that specifically cuts linkages in peptidoglycan. Hopefully you recall, peptidoglycan is the main component of bacterial cell walls. Lysozyme cuts those open so that the virus that made it can infect the cell. That would indicate to you in an indirect way that if a virus makes lysozyme, it is a virus that has evolved to parasitize bacterial cells. Another class of enzymes that are key and very important to some viruses are nucleic acid polymerases. So once again, this would be an enzyme that makes a polymer of nucleic acid, so something like DNA polymerase or RNA polymerase. There are some instances, in many instances, where viruses simply use the DNA polymerase or RNA polymerase that the host already makes. In other cases, the virus needs to make its own. A good question to ask yourself is, why would a virus need to make its own? If the whole idea is that it's really leeching off the host and just using the host machinery, why does it need to make its own? Well, if you are an RNA virus and you have an RNA genome, is there any genome in a host cell that is going to read your RNA and transcribe RNA out of it? There isn't. If you are something called a retrovirus and you have an RNA genome and you want to integrate into the host genome, you have to have DNA to do that. Human host cells do not have an enzyme that's going to read RNA and make DNA out of it. Normally we do the opposite. We read DNA and we make RNA. So if the viruses want to do those activities, they're going to need to have their own polymerase to do it. Another example would be things like neuraminases. So these are enzymes that cut a self-surface linkage called sialic acid. Flu viruses, again, hopefully everybody has seen that animation, they attach to sialic acid on host cells. That's how they bind and absorb and invade. Flu viruses leave the cell by budding through the membrane. If you have a flu virus that buds through the membrane, sees a sialic acid molecule, and immediately binds right to it, that viral particle is just going to be stuck to the cell it's already infected. It's not going to be able to transmit and infect a new cell. So if it has an enzyme that will cut away any sialic acid it happens to be stuck to, that cell is going to be much more transmissible than a cell that doesn't. I bring particularly these two up because they are the basis for some extremely potent and successful antiviral drugs. Just to give you one example, this one right here is a drug that some of you may have even taken within the past few months. A neuraminidase inhibitor that we should all be familiar with is this drug right here, Tamiflu. To a lesser extent, there's also its less famous cousin, Relenza. So these guys are drugs that specifically bind to viral neuraminases, prevent them from working, and so that prevents, in this case, flu viruses from replicating within your lungs and your nose. Okay, so to look at the host-virus relationship for a second, you have to have host cells in order for a virus to replicate itself. Okay, we've already said that. The reason being, now that you've seen what components a virus has, it has a capsid, it has a bit of nucleic acid, and it may have an envelope around it, but maybe not. There's nothing in here that's going to help this entity replicate itself. So it absolutely has a requirement for a complete cell in order to do that. Viruses tend to be very host-specific. As we talked about, they have a period called adsorption, where they pick out a host cell and they bind onto it and they stick. In the flu virus animation, again, they discuss this lock and key mechanism, or receptor and ligand, if you wish to talk in better biological terms. So the implication there is that every virus is particularly co-evolved to bind to a certain host. One last thing that would be also important to mention is that if you want to grow viruses, as I just mentioned, and now have mentioned several times, they don't replicate themselves. They need specific host cells to grow, and so if you want to grow some viruses in the lab, you can't just throw them into broth the way you would bacterial cultures, or you can't just streak them on a plate, as you would have done in intro micro lab. What you need to do is actually find a way of growing host cells in the lab, and then you infect them with virus. And in the process of doing that, those host cells are going to be the perfect vehicle for your virus to begin to replicate. Okay, so just want to point out two last quick things, that there are two different viral life cycles. Lytic viruses, or lytic infections, are what we've already been more or less talking about in that very undetailed introductory schematic. This is the standard path, where you have a virus, it infects a cell, it invades, its components are replicated, they're reassembled, and then they burst out. Remember, we called this process lysis. That's where you get the term lytic. Lysogenic infections are a little more complex. You have a latent phase to that. So you have a cell, it's infected by a virus. Instead of the viral nucleic acid replicating itself and then having its proteins expressed, what happens is that nucleic acid just integrates into the genome. And then as this whole cell replicates, it's quietly, silently, in a very subtle way, replicating that virus, because every time this chromosome is replicated, that viral sequence is replicated. If there is some particular induction to do so, such as the host cell is stressed, starved, immunosuppressed, whatever, this viral nucleic acid is going to take that as a signal of maybe it's time to bail out of here, because this host isn't doing so well. So this excises itself from the genome, and then it goes and enters this phase of life. It becomes lytic at that point. So the nucleic acid is replicated at a high rate, actively this time, not just subtly as a secondary effect of cell division. Its proteins are expressed, then you get viral packaging and assembly, and you get lysis. These guys are then free to go infect another cell, and then they start this process all over again. So fundamentally different from lytic infections. It's important because you can have maintenance of a viral infection for a very long time that is not being resolved either therapeutically or by the immune system. It's completely flying under the radar. This is critically important in human health. One of the most famous instances of this is HIV. Another real famous instance of this is human herpes virus. Another one is Epstein-Barr virus. Another one is chickenpox virus. So there are a lot of pretty big pathogens that undergo this process, and it really causes a bit of a problem in terms of their immunostimulation and their subtlety and their tendency to fly under the radar.